Hello, friends. Once again, one Saturday again, we continue with the biology of plants. Now we are in the chapter number 3. We continue with the mitochondria, this part of the cell of the plant that is really interesting as any other. Mitochondria. Um, if I have some question regarding the pronunciation, then I use my translator as always. So let's continue. Mitochondria, singular mitochondrion, like plastids, are surrounded by two membranes. The inner membrane has numerous invaginations. Okay, this Google Assistant is interfering with my voice. So the inner membrane has numerous invaginations called cristae, singular crista. The cristae occur as folds or tubules, which greatly increase the surface area available to proteins and the reactions associated with them. Mitochondria are generally smaller than plastids, measuring about half a micrometer in diameter and exhibit great variation in length and shape. Mitochondria are the sites of respiration. This is the key part. What is mitochondria used for? Mitochondria are the sites of respiration, a process involving the release of energy from organic molecules and its conversion to molecules of ATP, adenosine triphosphate, the chief immediate source of chemical energy for all eukaryotic cells. These processes are discussed further in chapter 6. Most plant cells contain hundreds of thousands of mitochondria, the number of mitochondria per cell being related to the cell's demand for ATP. Besides respiration, mitochondria are involved in numerous other metabolic processes, among them the biosynthesis of amino acids, vitamin cofactors, and fatty acids, and they play a pivotal role in programmed cell death, the genetically determined process that leads to the death of the cell. Programmed cell death is preceded by mitochondrial swelling and the release of cytochrome C, which is normally involved in electron transport. See chapter 7. The release of cytochrome C appears to be critical for the activation of proteases. How is this? Let's check this. It's like proteases, but maybe it's not like this. Proteasas in Spanish.
Proteases.
Wow.
Proteases.
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 Proteases and nucleases. Enzymes or enzymes? Enzymes or enzymes? Let's check this.
Enzymes.
Proteases and nucleases, enzymes that bring about degradation of the protoplast. Mitochondria are in constant motion, turning and twisting and moving from one part of the cell to another. They also fuse and divide. Mitochondria tend to congregate where energy is required. In cells in which the plasma membrane is very active in transporting materials into or out of the cell, they often can be found arrayed along the membrane surface. immotile or motile? Immotile or immotile? Let's check this. For one or another reason, today I have much more questions than other days. I don't know if it is only a feeling. Most times, twinning and twisting, they also find very active. They often
Motile.
Motile. In motile, single-celled algae, mitochondria are typically clustered at the bases of the flagella, presumably,
Presumably.
You see, it's good to check. Presumably providing energy for flagellar movement. Mitochondria, like plastids, are semi-autonomous organelles. The inner membrane of the mitochondrion encloses a matrix that contains proteins, RNA, DNA, small ribosomes similar to those of bacteria, and various solutes, dissolved substances. The DNA of the mitochondrion, like that of the plastid, occurs in one or more clear areas, the nucleoids. Thus, as mentioned previously, in plant cells, genetic information is found in three different compartments: nucleus, plastid, and mitochondrion. The nuclear genome, el genoma, genome o genome.
Genome.
Genome. Neither of them. Genome, the nuclear genome, the total genetic information stored in the nucleus, is far larger than the genome the genome of either the plastid or the mitochondrion, and accounts for most of the genetic information of the cell. Both plastids and mitochondria can code for some, but by no means all, of their own polypeptides. Mitochondria and chloroplasts evolved from bacteria. On the basis of the close similarity between bacteria and the mitochondria and chloroplast of eukaryotic cells, it is quite clear that mitochondria and chloroplasts originated as bacteria that were engulfed by larger heterotrophic cells. See in chapter 12. These larger cells were the forerunners of the eukaryotes. The smaller cells, which contained and still contained all the mechanisms necessary to trap and or convert energy from their surroundings, donated these useful capacities to the larger cells. Cells with these respiratory and or photosynthetic assistance had the advantage over their contemporaries of energetic, self-sufficiently and undoubtedly soon multiplied at their contemporaries' expense. All but a very few modern eukaryotes contain mitochondria, and all autotrophic eukaryotes also contain chloroplasts. Both seems to have been acquired by independent symbiotic events. Symbiosis is the close association between two or more dissimilar organisms that may be, but is not necessarily beneficial to each. The smaller cells now established as symbiotic organelles, which the larger cells obtain protection from environmental extremes. As a consequence, eukaryotes were able to invade the land and acidic waters, where the prokaryotic cyanobacteria are absent, but the eukaryotic green algae abound. Now we're going to change the paragraph. Now we're going to talk about peroxisomes. Peroxisomas. How is this? What is the correct pronunciation of this new word for me? Peroxisomes. You see, this vocabulary is most of this vocabulary is completely new. It's brand new for me. But with the correct tool, you can start studying
Peroxisomes.
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 You see, sometimes it's just the word to put the stress. Peroxisomes, no, peroxisomes. So peroxisomas, I still continue without knowing what are peroxisomas, even in Spanish. But peroxisomes, also called microbodies, are spherical organelles that have a single bonding membrane and range in diameter from 0.5 to 1.5 micrometers. They have a granular interior which may contain the body, sometimes crystalline, composed of protein. Peroxisomes contain no internal membranes and typically they are closely associated with one or two segments of endoplasmic reticulum. Once, though, to have the origin from the endoplasmic reticulum, peroxisomes are now peroxisomes. Let's check. Peroxisomes, yes. Yeah, it's good to repeat once and again. One, peroxisomes contain no internal membranes and typically, no. Once though to have thought to have their origin from the endoplasmic reticulum. Peroxisomes are now known to be self-replicating organelles, like plastids and mitochondria. Unlike plastids and mitochondria, however, peroxisomes possess neither DNA nor ribosomes and must therefore import the materials required for their replication, as well as all of their proteins. Like plastids and mitochondria, peroxisomes have been shown to undergo movement within the cell. Some peroxisomes play an important role in photorespiration, a process that consumes oxygen and release carbon dioxide. Exactly the reverse of what happens during photosynthesis. In green leaves, peroxisomes are closely associated with mitochondria and chloroplasts. Other peroxisomes called glyoxysomes contain the enzymes necessary enzymes necessary for the conversion of stored fats to sucrose during germination in many seeds. The two types of peroxisome are interconvertible. Okay. We continue a little bit more. We are in 40 minutes. Maybe we even finish the vacuoles. Let's see. So we start with the vacuoles. 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 So vacuoles. Together with plastids and a cell wall, the vacuole is one of the three characteristic structures that distinguish plant cells from animal cells. As mentioned mentioned previously, vacuoles are organelles surrounded by a single membrane known as the tonoplast or vacuolar membrane. The vacuole may originate directly from the endoplasmic reticulum, but most of the tonoplast and vacuolar proteins are derived directly from the Golgi apparatus, which is discussed on pages 52 to 54. Many vacuoles are filled with a liquid commonly called cell sap. It's very curious because the vacuoles are very huge. Makes, let's say, that use a big space in the cell of the plant. What are these vacuoles consist of? So these vacuoles are many of them are filled with a liquid commonly called cell sap. The principal component of the cell sap is water, with other components varying according to the type of plant, organ, and cell, and to their developmental and psychological states. What? Developmental? Maybe, but physiological. Psychological is impossible. So it's physiological states. Okay. In addition to inorganic ions such as calcium, potassium, chloride, sodium, and phosphate, vacuoles commonly contain sugars, organic acids, and amino acids. Sometimes a particular substance is present in such a high concentration that it forms crystals. Calcium oxalate crystals, which can assume several different forms, are especially common. In most cases, vacuoles don't synthesize the molecules they accumulate, but instead receive them from other parts of the cytoplasm. The immature plant cell typically contains numerous small vacuoles that increase in size and fuse into a single vacuole as the cell enlarges. In the mature cell, as much as 90% of the volume may be taken up by the vacuole, with the rest of the cytoplasm consisting of a thin peripheral layer closely pressed against the cell wall. By filling such a large proportion of the cell with inexpensive vacuolar contents, plants not only save expensive in terms of energy, nitrogen-rich cytoplasmic material, but also acquire a large surface between the thin layer of cytoplasm and the protoplast's external environment. You see? So the vacuoles are, let's say, like inflate rubber inside the plant just to extend physically the parts of the plant. In this case, the leaves. Most of the increase of the size of the cells results from enlargement of the vacuoles. Yes. A direct consequence of this strategy is the development of internal pressure and the maintenance of tissue rigidity, one of the principal roles of the vacuole and tonoplast. Exactly. Different kinds of vacuoles with distinct functions may be found in a single mature cell. Vacuoles are important storage compartments for primary metabolites. Metabolites or metabolites? Always some question. This metabolitos. What's going on here?
80% de batería. Conectado a Galaxy Note 9 de José Luis.
Metabolites.
Metabolites, you see? Metabolites, no. Yeah, something is like it's always the same. You used to do in certain way, and then it's not that way. But you are suspicious about that. You say, okay, something is wrong here, but what is wrong? It's always the same. Metabolites. For primary metabolites, such as sugars and organic acids and the reserve proteins in seeds. Vacuoles also remove toxic secondary metabolites such as nicotine, and tannin from the rest of the cytoplasm. Such substances are are sequestered permanently in the vacuole. So the toxic substances are also stored in the vacuoles, like let's say, like something similar to the animals with the fat. In the fat, we let's say storage all the toxic metals and in general elements in our body in the fats. So such substances are sequestered. Let's check this. Sequestered. It's a strange word. This embargado, secuestrado, embargado.
Sequestered. Sequestered.
Sequestered. So are sequestered. Such substances are sequestered permanently in the vacuoles. As discussed in chapter 2, the secondary metabolites contained in the vacuoles are toxic not only to the plant itself but also to pathogens, parasitos, parasites. Oye, oye, oye.
Parasites.
Parasites. So pathogens, parasites, and or herbivores, and they therefore play an important role in plant defense. The vacuole is often a site of pigment deposition. Pigment deposition also. So these toxic areas or this toxic content storage in the vacuole prevents the plant from the, let's say, parasites, pathogens, and herbivores. Oh, curious. But also is where is a plant deposition, also color, pigment deposition, the blue, the violet, the purple, the dark red, and scarlet colors of plant cells are usually caused by a group of pigments known as the anthocyanins. And like most other plant pigments, the anthocyanins are readily soluble in water and are dissolved in the cell sap. Okay, so this kind of pigments are also in the vacuoles. Very, very amazing. Mm. Mhm. With the group of color blue, violet, purple, dark red, and scarlet. They are responsible for the red and blue colors of many vegetables, radishes, turnips, cabbages, and fruits, grapes, plums, cherries, and a host of flowers, cornflowers, geraniums, delphiniums, roses, peonies. Sometimes the pigments are so brilliant that they mask the chlorophyll in the leaves, as in the ornamental red maple. In the ornamental red maple is more important the, let's say, this pigment that the chlorophyll. Anthocyanins are red colors of some leaves in autumn. Okay, so in autumn, these colors turned to brilliant red colors in autumn. Yes, it's normal because the sunlight is dropping, but still the pigments are there. So that's the reason it still looks like red. No green, of course, but red. These pigments form in response to cold, sunny weather when the leaves stop producing chlorophyll. You see, it's the first time I'm reading this book, but I think that I'm understanding quite well. As the chlorophyll that is present disintegrates, the newly formed anthocyanins in the vacuoles are unmasked. In leaves that don't form anthocyanin pigments, the breakdown of chlorophyll in autumn may unmask the more stable yellow to orange carotenoid pigments already present in the chloroplasts. Normal. So if we don't have these pigments, then appears another pigments like yellow to orange carotenoid pigments already in the chloroplast. So if you have a leaf with is green normally, and the color of a leaf is green in summer and in spring, but then in autumn this green turns to yellow to orange. It's normal because chlorophyll disappears or get down, and then only remains the carotenoids that has this orange color. But in some other plants, there is a question of the pigments in the vacuoles are red, violet, and etc. And can, let's say, be over this, let's say, can be stronger than the orange effect. And then you can see like light, like a red color. So the most spectacular autumnal coloration develops in years when cool, clear weather prevails in the fall. So the most spectacular autumnal coloration develops in years when cool, clear weather prevails in the fall. So if in autumn we have sun, cold, then we have the red, strongest red color. It's very, 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 very curious. Vacuoles are also involved in the breakdown of micromolecules and the recycling of their components within the cell. Entire cell organelles, such as mitochondria and plastids, may be deposited and degraded in vacuoles. Before of this digestive activity, vacuoles are comparable in function with the organelles known as lysosomes that occur in animal cells. Very, very curious, these vacuoles. I enjoy quite a lot this part of the chapter number 3, the plant cell and the cell cycle. And friends, we are going to stop here. Please subscribe to the channel, and I hope you enjoy, you learn something, and of course, we see in 1 week. Please remember to subscribe to the channel, like if you like this kind of video. If you have some comment, please comment. And remember that we have 4 channels. We have Estrategias Diarias, Estrategias Diarias Ingeniería, Estrategias Diarias Cultura, and now a new channel, Estrategias Diarias Gold. Okay, have a nice weekend and see you very soon. Goodbye, friends. Goodbye. Enjoy your time. and enjoy the nature.